हेलो हेलो हेलो। टाइम नहीं जाएगा। हेलो 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 हेलो। जा रहा है, हमको सुनाई दे रहा है, आपको नहीं सुनाई दे रहा। हेलो हेलो। हमको सुनाई दे रहा है यहाँ से। हेलो हेलो।
Good evening to all of you. I welcome you all from this behalf of History of Medicine Society. And this is our second lecture in this series. And this society is scheduled to have monthly lectures. So this time we have none other than the President of Bacteriophage Society of India and our Dean Research, an eminent scientist, academician, and my beloved guru, uh, Professor Gopal Nath sir. Uh, he will be speaking on the topic of his interest and the one which he have been working since last like 30 years time. So we all welcome Professor Gopal Nath sir. And sir, from this time, we also have sir live YouTube channel with the society, and so that everyone, this history is same in all over world. So may it be America, may it be Australia, may it be India, history cannot be changed. So with this vision and view, we have this live channel. So let's hope that people will join in coming time, and later also with share, people will be seeing. So now it is already 4:10. We have like 40 minutes of sir's presentation. And after this presentation, we will have a quiz. And then after quiz, we will have a participation certificates. And so I request all of you to stay till the whole talk and the quiz. And we'll try to gather the knowledge. Professor Gopal Nath sir. So thank you very much. And actually, history of medicine is a great step we have started, and whatever we know since the beginning of the civilization, everything is history. Hello, hello. Yeah, yeah. ठीक है. हम धीमा बोलते हैं ना. So, if you want to make the subjects easy, so we used to tell the students that whatever is written in the book, everything is history. So don't take the mathematics and physics like a very tough subject. They are all history because the humankind have already done it. So, and for shaping the future, it is must that you should know the real history. So this step of the history of medicine started by IMS is a great step. And everything should be taken as studying history, not the microbiology or anatomy. It looks very tough, but if you see, they are all history. So with this, so I shall be speaking on this topic with special reference to strange history of bacteriophage therapy. And fortunately, it started from India only. And then the biggest clinical trial was also done in India. Very few people know about this. So I invited in 2017 one of the American scientists, and he first time informed that the biggest clinical trial was done in 1938 in Assam and the West Bengal, and they added the bacteriophage in the drinking water in different means selected villages, and the other were not given, and they found that this is effective. So for the if you see the complexity of the human body, so before going to the all these complex things, I would like to tell you that how difficult it is to still to remember these muscles, though they are multicellular usually. And if I ask that, I don't know, I may tell about 10-20 muscles today, not all. But even the difficult to this one is that if I ask that how many cells are in the human body, I'm just giving you the introduction of the microbiology. So the answer is this.
and total 30,000 genes are expressed from the human body, and total numbers are 10 power 13. So 4.5 years we used to teach about the body, physiology, anatomy, biochemistry, and microbiology is being taught only 365 days, that too maybe 200 days. But the number is this. What may be the approximate number of microbes in and on a human body? 10 times more than the human cells. That is 10 power 14. And the total microbe, if you say, that is 100 times more than the human cells. So that is 10 power 15. So our body is having 99 part microbes by number and 1 part only human cells. So I feel that the why not the microbes are so important. Now we are just studying about the gut microbiota and all those microbiota in the human health, and they are affecting because every cell is governed by 100 different microbes, and we cannot do anything by ourselves. If you take the sterile seed with sterile water and sterile soil, it will not thrive. The bacteria or microbes are essentially needed for the growth. So that is the fact about this. So we are having we know that these are the normal flora of our body and usually they are helping us, but sometimes they are causing problem to us. So we are having two extraneous factors to deal with the bacterial infections. One is antibiotic, another is the bacteriophage. And you know, the in nature, so the research can be better done by exploiting the processes which have evolved in the natural evolution, not by human effort that you are just trying to have the synthetic molecule that this and that is immediately taken over by the nature. So these antibiotics and bacteriophages are basically the natural process in the natural evolution process. Antibiotics are signal molecule to protect the bacterial biofilm at sub-inhibitory concentration. So basically they are destined to make the biofilm. Any single cell, any single body, maybe bacteria, maybe virus, everybody wants to make a cumulative effect that they want to make a family. That is biofilm. For survival, it is always better. And we are also making, we are IMS family here right now. Then bacteriophages, they are basically genetic engineer of the bacteria. So that whatever adverse conditions are coming, they are modifying genetically to have the hundreds and thousand types of variants, and any of the variants will be able to survive the harsh condition. That is the basic thing which has been already present in the nature. So how we got the antibiotics first time? That by random observation that there are certain molecules which are inhibiting the bacterial growth, maybe produced by the fungus, and then bacteriophages I shall be telling you. So these are the for the UG. Actually, I am having this. So just mnemonics for this: antibiotics acting on cell wall, antibiotics acting on the cytoplasmic membrane, antibiotics acting on this protein synthetic machinery. If you want to remember, there is the mnemonic, and then the antibiotics acting on DNA and on RNA, and this is on the anti-metabolites in the cytoplasm. So these are the total groups because there is difference in the structure of these cells of the bacteria than to the human being. So we are exploiting that there are certain molecules which are going to affect selectively to the bacteria, but sometimes they are also affecting the human cells. Like all these molecules which are acting on the cytoplasmic membrane, usually they are cytotoxic. Cholesterol is one of them. Triazoles, polyenes, polymyxin B, and polymyxin E. You know that they are renal toxic because they are acting there. But none of the antibiotic is safe. They somehow, somewhere, they are acting on our cells also. So there are a total of 26 different molecules, and you can see that from where they derived. Actinomycetes, other bacteria, fungal origin, and synthetic. These are the total classification of the antibiotics. Synthetic molecules, though we have, but they are having, means they are not very successful because the bacteria do develop the mechanism very fast against the synthetic molecule. So this is the evolutionary means development of the different antibiotics, and you can see first time we started using antibiotic penicillin in 1940. Next year we got the resistance development, and majority of the antibiotics you can see were developed in 1940 to 1970. Later, we are having very few molecules, and pipeline is almost dry. For taking any antibiotic molecule in the clinical practice, it takes 20 years. All those in vitro studies, preclinical studies, clinical trials, then only it is coming to us. So 20 years is a long time. And bacteria develop the antibiotic resistance on the next day. So this is this story. And this is why we have to bother about this. But most important is that we started using most of the antibiotics in 1940 or onwards. Only some synthetic molecules like sulfur drugs were used earlier. But before that, if anybody is having the infection, what was the fate? Before 1940, any First World War, any injury, staph aureus infection, they soldiers have to die. There was nothing with us. Even the first time penicillin was used, one of the soldier, but he died because there was no enough dose. So that has happened. And the antibiotics, the bacteria are very smart, very, very smart than the human being. And then that's why they are having all the mechanism, whatever we are applying. So we must have and we have to make the balance between the two. So, but before that, what was happening? People were just looking for something that can help us. So that started from in India only. So you can see this is the journey of the antibiotics and end of the road. Very few molecules have been added later on after 1980. And there are bacteria which are resistant to all the available antibiotics. This is the fact. And we are watching patients dying in the ICUs and many places. So there was a surgeon. Surgeon usually, sir, Professor Shukla is sitting here, so please don't mind. Surgeon looked with the different perspective, and the microbiologist looked with the microscope. So what he said in 1965 and 1969, because so many molecules were there, Dr. Stewart, the United States Surgeon General, is primarily known for his legendary statement, "It is time to close the book of infectious diseases and declare the war against the pestilence won." But is it fact today? It cannot be. The war can never be won by anybody, any this army of these things, bacteriophages, bacteria, or human being. It is continuous process. We have to look together. Basically, we are beneficial to us. Sometimes they are causing. So basically, we have to control. We need not to eliminate them. So war is always going on. And the same thing was said by other one also. So you can see the death because of the antibiotic resistance in 2019. Antibiotic resistance is directly causing death in 12.7 lakh people globally. And death associated with antibiotic resistance, 49.5 lakhs. HIV AIDS, 8.6 lakhs. Malaria, 6.4. So this is the extent or magnitude of the antibiotic resistance which we are facing today. So science starts with this. I was just talking to Dr. Veer Mishra that we should not, since the Enlightenment, scientists have cultivated a self-image as rational actors guided solely by reason, evidence, and logic. So in science, there is no scope of astha or bhakti. Forget about this. If somebody is telling, don't believe it. You have to test. You have to test at several times at different locations. When the consistent result is there, then only you can say that this is effective. So when the Royal Society of London was founded in 1660, it chose as its motto "Nullius in verba," often translated as "on the word of no one." So we are not going to believe anybody with our closed eye. Referring Horace Epistles, "Nullius edictus irure in verba magistri," it means being not obliged to swear elegance to any master. हिंदी में कहते हैं कि कोई भी बोले और सर झुका के मानेंगे नहीं जब तक कि हम उसको रीजन, एविडेंस एंड लॉजिक से प्रूव ना करें. If somebody has said, don't believe it. If it is in tradition, don't believe it. If it is written in the book, don't believe it. You have to test, we have to test. When we are getting the consistent result, that is the science, then only we have to believe it. So this is the time, and we are just following in the history of medicine. Similar to our 21st century contemporaries who embraced the new evidence-based medicine, Dr. Shank spoke on that day, but the medicine should be in the right perspective, right context. It's not that somebody is telling that this is my evidence-based medicine and we are believing it. No. A virtuous of the Royal Society heralded a new era in science based exclusively on observation and direct experimentation experience. So this is the basically discovery of bacteriophages. If you know, there was a scientist Hankin, and he was just traveling across the Yamuna and Ganga. He found that the people who are drinking the river water, there was less mortality, less morbidity due to cholera than the people who are staying far away and not consuming the river water. So he concluded, because of many regions, that he found in Agra that there was reduction downstream, there was reduction in the count of the Vibrio cholerae than in the city. So that there was a huge number of the Vibrio cholerae contamination, but after that there was cleaning effect because of the bacteriophages. So basically it happened in 1996. He writes Ganga and Jumna, and he found this one, and from here somebody thought that this might be there might be something smaller than the bacteria. Before 1880, nobody was believing that there are small structures which are 1 to 2 micron in size. So there was no microbiology department. There are so many people sitting, and the person who the surgeon operating this one, they were not knowing that why their patient is dying, why tuberculosis is killing the patients. There was nothing with us. It was the microscope. That's why I was telling that we should have a talk on the history of microscopy also, because that is the basic issue which has given the different perspective to the whole medicine. और हिंदी में कहावत है, नजरिया बदल दीजिए नज़ारे बदल जाएंगे. अगर आप माइक्रोस्कोपिकली देखने चलिए तो आपको लगेगा कि दुनिया डिफरेंट है. तो दैट इज द वेरी बेसिक कंपोनेंट ऑफ द मेडिसिन along with so many important things. So let us see the history of this one. The bactericidal action of the waters of Jumna and Ganga in Vibrio cholerae, he writes like this. Ernest Hunbury Hankin, in 1896, he found water source Jumna River near Agra 10 power 5 per ml, and 5 kilometers downstream there was the count of 10 power 2 only, and there was a reduction in the Vibrio cholerae count downstream, and some factor was acting, but there was a lot of history about behind this. We'll come to know about this. The bactericidal action of the water of the Jumna and the Ganga on Vibrio cholerae, and experiment he did was that he mixed the filtered water in the bacterial growth of the Vibrio cholerae, and he found that there was reduction in the number in vitro also. So he concluded that there is something in the water. We Indians, we are the Jagat Guru and we are the Vishwa Guru, but all these findings have been observed by the outsiders only. Why we are not doing, I don't know. So this is the fellow taught. He observed glassy, clear areas in the colony of Staphylococcus. Could not be cultured independently. It was growing only with the bacteria. It could be serially transferred and propagated on new bacterial colony, but not on dead bacteria. So they were thinking that this might be amplifying enzyme, but why it was propagating along with the bacteria only? He could observe, but he did not pursue for long this research. But because he was British, so there are two groups. So Britishers were having the upper hand at that time in the beginning of the 20th century. So every finding, everything they were propagating that they have found it out. They were not giving the credit to the Germans and French and Japanese and the Russians. So that also happened with this. And this antibacterial activity was destroyed by the heat. So it was heat labile. Then this fellow, self-trained, that is called as autodidact, self-trained microbiologist, not bothering to the whole world, whatever he was thinking, he propagated that he is always right. And his contribution is the maximum. He never even bothered for the Nobel Prize. His all contemporaries got it, and the major opposition came from all those Nobel laureates. And he told that I'm just keep on working. And today we are remembering this Felix de Herel, the maximum about the bacteriophage. Or I think these man-made prizes and all these things are not that important. We should work for our satisfaction. So what anybody will give? You make a company and you just tell me that I'm making you this and that. It doesn't matter. Like recently we thought that we found that there is a scientist from chemistry and telling that the Ganga water is sterile at Kumbh. It is not possible. So we must contest it. Nobody spoke against this. So this 1919 to 1930, the pre-antibiotic era, from its prophylactic use against avian typhosis, the D. Harrell used and he found the results, but the results were taken as controversial. Some saw it was the panacea for all infectious diseases, while others thought it was oversold and probably worthless. So there were two schools of thought. One were thinking that this is really very effective, another was telling that this is not effective. And you must understand that microbiology at that time was at the very preliminary stage. We are not knowing, just watching a gram-negative bacteria, we are thinking that all gram-negative bacteria rod-shaped are same. Even E. coli has got the thousands and thousands of the variations. We have given the E. coli name, but E. coli are not same. So that time we are not knowing that they are gram positive, they are different, and there is strain variation. Even Klebsiella pneumoniae, by our identification, one bacteria from one patient is sensitive to that bacteriophage. The next bacteria of Klebsiella pneumoniae is not that is not getting killed by the bacteriophage. They were not knowing at that time. So while it is no doubt true that the discovery and widespread availability of antibiotics in the immediate post-World War era undermined the enthusiasm for the fast therapy, that is not the whole story. It is not that the broad spectrum activity without necessity of knowing that which bacteria, which bacterial strain is causing the infection. हनुमान जी के संजीवनी बूटी के पहाड़ की तरह लेके आए, उसमें से निकाल के दे दीजिए. Any antibiotic you give, even penicillin was so effective against the so many means bacteria. Simple crystalline penicillin, I remember during our childhood. So it was that. That's why the so specific smart bomb-like bacteriophages were undermined. It is indeed true that the fast preparation did not have the first convenience of the antibiotic. Antibiotic never bother what bacteria is there. It is the bacterial infection. If the physician is convinced, he will give and he will get the cure. Then we are not broad spectrum. Bacteriophages are never broad spectrum. But we can try to find out some of the broad. We are doing that one, but that will take further a lot of time. And suffered greatly from the scientific controversies on the biological nature of phages that played out in 1920 and 30. So there was the confusion whether they are the simple molecules or some structures smaller than the bacteria. Nobody was believing that anything will be there which is smaller than 1 micron and that live too. Viruses we could be convinced in 1945 after the discovery of electron microscope. Before that, we were not knowing that there are viruses. We are all the time talking about corona, AIDS, this, that. So it is not long away. Very means in the recent past we have got it. But what the other more subtle reason for this decline, we have to find out. This is the fellow. And he was very close to the Fage Church group. What is Fage Church? Fage Church refers to the informal group of scientists including Max Delbrück, Salvador Luria, and Alfred Hershey. You must have heard the Luria and Hershey experiment, Delbrück's experiment. So they were used the bacteriophages as a genetic tool for so many things, and they really give very significant findings. But they were sure that bacteriophages cannot be used in the therapy, and he was one of the close of that group. His name was Stent. A key figure of Max Delbrück's Fage Church provides a compelling argument against the effectiveness of fast therapy. And in 1963, he wrote a monograph. What he writes? First, he suggested that it advocates very scientifically sloppy. Many physicians manage to convince themselves, clearly implies that These physicians even referred to as converts, suggesting religious than the national analyst, were guilty of wishful thinking rather than clear-eyed scientific judgment. He's telling that they were just having the astha-based belief that the phages are really working, and there was no scientific reason, no logic. Second, हिंदी में इसका अनुवाद है, सबसे पहले उनका सुझाव है कि इसके समर्थक वैज्ञानिक रूप से लापरवाह थे। कई चिकित्सक खुद को समझाने में कामयाब रहे का स्पष्ट अर्थ है कि ये चिकित्सक यहाँ तक कि धर्मांतरित भी कहे जा सकते हैं, जो तर्कसंगत विश्लेषण के बजाय धार्मिक विश्वास का सुझाव देते हैं, स्पष्ट वैज्ञानिक निर्णय के बजाय इच्छाधारी सोच के दोषी थे। इनके एक एक शब्द पे you have to just. So that was the story that time, and he wrote in a book. Second, stents associate phage therapy with the medicine of the American World War II enemies. See, he connected the phage therapy with the World War II enemies, and who were they? Italy, France, Japan, and USSR. And then phage therapy was somehow taken as un-American. So anything un-American was not accepted by the West world. Third, even today, phage therapy persists, suggesting an outdated technology found in out of the way places. So out of the people who are practicing the phage therapy, they are out of place people. So that these three things was propagated by the stent. With the authority and not a little humbles, stent proclaims that chapter to be fairly considered closed, as that surgeon told in 1969. He also tells in 1963 that this chapter should be closed. This is the part of the history. But history is having so many things. If you are doing really good work at the Institute of Medical Sciences, so there will be very many enemies. Whatever he's saying is wrong. And they will be just telling on the personal basis. They'll be telling on many other ideological basis, geographical basis. And all these divisions are there. This is the human mind. They will keep on doing this. So in both the cases, you can see in the antibiotic also, in this case also, somebody suggested that now the book should be closed. So Stent's verdict on phage therapy had its root in the early controversies in phage research. There was some problem, the lack of understanding of phage biology. Now we know a lot about the phage biology. At that time we were not knowing. The inherent difficulties of evaluating therapy in human clinical trials. So the concept of clinical trial also came very late. We are now having the randomized double-blind placebo-controlled trials. But at that time, people were not thinking, they started thinking that that should be done. This history is as personal as scientific. From the very discovery of phages, this field has seen personal attacks, disputes of priority, massive egos, and international politics. Even when I started working in IMS about the phage therapy, I got a lot of personal attacks. I'm talking of 2005, 6, 7, 8, telling all these is fail and this fellow is just unnecessarily doing all this research. All of these are the part of the rocky history of phage therapy. The very discovery of phage was the subject of prolonged dispute between the two camps. One camp was Felix de Herel, French Canadian. French plus Canadian, not American, not British. Autodidact, who was the first to identify bacteriophages as virus that infect bacteria, aligning with our current understanding. This was the person who first time said that they are the smaller living structures than the bacteria. In 1916, while working as an unpaid volunteer at the Pasteur Institute, de Herel discovered crucial facts about the bacteriophage that observed in the filtrates of dysentery fluid. And luckily, you'll be happy to know that de Herel visited thrice India. The British government at that time invited him to control the plague and the cholera, and also in Bombay he was invited for some probably animal this one, and he tried there. On the opposing side were the supporters of Frederick Twart, a distinctive British microbiologist known for his observation of transmissible grassy transformation in bacteria. However, Twart did not pursue further research based on his initial findings. In the early controversies surrounding his work, he was represented by an unexpected advocate, the Nobel laureates. So opposite side, there were so many noble laureates. And this poor Felix de Herel was alone. But he was fighting. Somewhere he was wrong also, making statement. This is Jules Bordet. You might be knowing about the discovery of the Jules Bordet. He first time invented the antibodies and told that the bacteria are lies by because of the antibodies. So he told that the bacteriophages are doing nothing. It is the antibody which is lying. It might be certain enzymes which are doing this job. So Joule got the Nobel Prize for his work on immunity based on the lysis of bacteriophage antibodies, not phages. Undaunted, the iconoclast de Herel, in his second monograph on phage, boldly challenged the famous Bordet, director of the Pasteur Institute in Brussels, by describing Bordet's work as the history of an error. But he was wrong here. It was antibody do this. So because there were two groups, opposing groups, so that's why he made this statement. This affront did not go unnoticed. Bardet and his protege, Andre Gracia, launched a nearly decade-long campaign against de Herel and his research. They disputed de Herel's view of bacteriophages as viruses, believing instead that they were merely induced lytic enzymes. There is nothing like living molecule. Additionally, they claimed that Frederick Schwartz had made his discovery first, challenging de Herel's priority in the field. So they are not ready to accept the priority or de Herel as number one. This is another issue. This is human nature. De Herel strongly contested the prevailing views that he lacked significant institutional support because the Nobel laureates were not to his side. Our subtle approach. In 1931, he resorted to the court in Paris to compel the editor of the Annals of Pasteur Institute to publish his challenge for scientific duel against Bordet. Surprisingly, the majority of scientific community at the time, including all the standard textbooks, supported Bordet's idea of phage as a self-perpetuating lytic enzyme. Because Nobel laureates were telling, so that's why it was true. In India also, please don't mind if Jagat Guru and the Vishnu Guru are telling that that is the right thing. We need not to test on the scientific ground. We should not, we must not. So another Nobel laureate was John Northrop, the Rockefeller Institute, and a winner of Nobel Prize on his work on digestive enzymes such as trypsin and pepsin. His authority to the lent his authority to the idea that phages was another example of autocatalytic self-activation similar to the trypsinogen and trypsin and pepsinogen and pepsin enzymes. This view of phage as lytic enzyme was strengthened by the support of another Nobelist who took up phage research in 1930s. Now the electron microscope comes in. It was probably 1932 when it started, but first time in 1939, the photograph of the phages was taken. Now they had to believe that they are smaller structure than the bacteria, and it was done in Germany. So the Ernest Ruska was the scientist who first time used the electron microscope, where the magnification can be done 1 lakh times, and the resolution power is 0.2 nanometer. Very small things can be seen. So from 1930 to 1945, in these early days, standard method and materials substantially controlled trials and double-blind studies were nearly unheard. That's why the validity of the phage research or phage therapy could not be done. Instead, small trials, anecdotal reports, historical controls, and clinical impressions were the norm. It is small wonder then that controversy arose. Definitely it would be. One approach to resolve the ambiguity was attempted by American Medical Association, and they published 3 different papers. So initial 2 papers rejected the idea that the bacteriophages are the basically living molecules. But the third report, which came in 1945, they agreed and they were convinced because by that time the electron microscope demonstrated that this is the structure of the bacteriophages. So they emphatically supported and the second report they did not support, they went with the Bardet. And the third report, only 4 years after Kruger's and Scribner's, in a complete reversal of prior dogma, fully accepted the viral nature of phage. Indeed, a few electron pictures were worth many thousands of the words. And by that time, the virology also started with the advent of the electron microscope. Big pharma companies made huge profit with new antibiotic molecule, the wonder drugs, but now we are sitting at the verge of revisit of pre-antibiotic era due to emergence of PDR bacteria. The basic nature of phage as a smart bomb is another issue because they are too specific. And the shelf life of phage preparation is another issue to be addressed. So it is very important after preparation to the delivery to the patient. Stability is very important. Cold chain should be maintained, and then whether it is really and customized phages are the only answer. So with this, if you see, so there are still some problem, but a lot of has been done. So I'm just showing very few of my work in coming 5 minutes only. So this patient is from Hyderabad, and then we treat these patients on the compassionate ground, and he was have developed the ulcer on in May 2023, and we took the consent, and then he visited here to our lab, and this was the initial position. And surgeons, they were telling that they have to go for amputation. And that fellow, a technician in the radiology, was not accepting that I will be having without leg. So then we started the therapy, the bacterial staph aureus, we prepared the phages and we started, and you can see the progress. And it was having osteomyelitis also. You can see the bone chip there in the middle of the wound. And this is on the 41st day. Again, you can see the bone. So I said that the bone can be removed, but no surgeon was ready to remove there at the Hyderabad. Then I said, you come here. And then I just discussed with the Dr. Vivek Srivastava. He told that I will do. And then they came with the whole family, 5 people by flight, not very rich people. And when they went to the ward, the bone itself came out. And you can see the spur formation showing the osteomyelitic changes here in his foot. And I just put this, you can see this is there. This bone chip is here. That happens in our hospital only. And he was convinced that only BHU can do something, nobody else can do in India. So he was so confident. And then you can see 120th day. So we started from here and this ends here. That is the magic of the bacteriophage. Another very quick patient was in septicemia because of the implant at the hip joint, and she developed the Klebsiella pneumoniae septicemia, and the Klebsiella pneumoniae was resistant to cholesterol also. So no drug was available to treat her, and her procalcitonin level went to 5.25. Her total count was 29,000, and then the polymorph count was 87%, and the CRP was 240. Blood urea was 125, and the creatinine was 3.5. So I was not knowing the basic course of this infection, but one day I asked one of the anesthetic friend. He told that 3 or more days she could survive in this stage only. So then his son is a scientist in DBT, and one of his his office person, he learned about the whatever I speak in the Chennai conference, national conference, and then he said that there's something can be done. The patient was admitted to the Apex hospital only, and he contacted us, and then we started, and she was having the history of, she got the injury 2009, and two more injuries they were there, and these are the parameters which I was telling. CRP, you can see the total leukocyte count and the polymorph count. And then we took again the consent from the patient. And then we started therapy on 2nd December 23. And on 3rd, these are the viruses by electron microscope. And this we could do because there was a PhD thesis done in my department. And then the sequencing was also done. We were having the pure endotoxin free, good manufacturing practice cocktail of 3 broad spectrum phages with us. So immediately we gave it, and patient could revert in 3 days. This was the 5th day. Total leukocyte count remained the same. That was a bit lower. There was scanty pus discharge. Fortunately, all the 3 phages in cocktail from the wound were lytic to the infecting bacteria. But when the bacteriophage, the Klebsiella came down, another bacteria appeared in the huge number, that was Robacter. So it seems that one bacteria inhibit another bacteria. There may be mixture of the bacteria usually in such cases where biofilm formation is there. So then we took the Acinetobacter baumannii also. We prepared the phages. Already phages were there. We just prepared on this. This is the wound condition on 7th December. And then on day 7, you can see the procalcitonin level came down normal. And then hemoglobin again rose, blood urea came down 36, serum creatinine 1, CRP is still high 71, but this is much lower than 240. The new isolate could be confirmed abnormally, and then we tested 10 purified bacteriophage against it, and then again we gave this one. When the blood parameters came down, the surgeon decided to open the wound, and we were simply irrigating the side with the bacteriophage cocktail. So because time is not that much, so I'm just rushing. You can see day 14, and then we kept an eye again. The Klebsiella kept on coming when the treating surgeon closed the wound, as the blood parameters were normal. Now it came down 8,000, 9.1, and CRP was this, but still bacteria were growing. Klebsiella pneumoniae was growing there, few colonies only. And then the drainage tube were put, and we are pushing the bacteriophage cocktail through the tube inside the wound. So day 20, you can see this is the tube, drainage tube they were having. And you can see how much discharge is coming like this. And this is the wound condition. This is the collection is still sterile fluid to 120 to 150 ml coming daily. And then you can see all these parameters are normal now. But we kept on giving the bacteriophage up to 152 days till 7 days before this wound site became totally sterile. And then this is the wound condition. I shall be showing you last video only. Seven wound swab collected on alternate day were found sterile, and we stopped the fast therapy on 17/4/24. We started the journey on 2nd December. And then this is the, now she can walk. She was having very healthy wound, and she is all right. Otherwise, no antibiotic was working. If his therapy was not there, she was not going to survive. And this was the course. So with this, I think I spoke more than 40 minutes. Thank you very much for your patience listening. This is the history of the bacteriophages, and thank you very much.
I think we have just listened one of the historic talks, sir, with so many Nobel laureates in one talk. I have yet to find any such field that maybe more Nobel laureates can come and join the series. Sir, since we are having very short of time, so we'll have a quiz, and before we end up, so quiz, one thing that the students are there to distribute the papers. We have 6 minutes to you, 20 questions. 6 minutes, 20 question. Topic is bacteriophage. So we'll start the time once it is all distributed. So can you please distribute all the papers? Give it here. So please keep your mobile upside down, and the invigilators are next to you. One have invigilator next to you. Yes, sir also. Sir. YouTube को बंद कर दो. So match. So you have to match. उधर मिल गया सबको? Please give it. So we start 5 minutes after you get on the back seats. So we start from now. Total 6 minutes of quiz. 6 minutes. Maybe 17 seconds per question. Damni, dekh time. It reminds us our school time, college times. Dr. Naveen, solve your one. Please, no, not Tino. No Tino, एक का दीजिए सबको. No Tino. पेन ले लीजिए, येलो पेन लो. Already you have lost 1 minute. You are all examiner with each other. Please keep your mobile upside down and then write your name of your on your corner, upside corner, name and department. If you're a student, please write your left. So 4 minutes left. No negative. Yeah. Excuse number.